Okay, so yesterday I'm going to talk about the PhD work that I did with Corinne at King's College London, is this going to work? Yes, where I studied FOXG1 syndrome, which is a rare neurodevelopmental disorder caused by heterozygous mutations in the fork head box G1 gene. The gene itself encodes a transcription factor that has roles in just about everything during the development of your telencephalon. And the disease itself really ranges in severity with a strong genotype-phenotype correlation, whereby mutations that are frameshift or truncating in the N-terminus tend to be the most severe. And by that, I mean the children can't walk, they can't talk, they can't even sit up unassisted. So, with that in mind, our zebrafish model of the disease really reflects the most severe human condition, whereby we have this 5 base pair deletion causing a frameshift and the induction of a premature termination codon, which should in theory mean there's no protein produced. So my first aim was to characterize this mutant and try and establish it as a reliable disease model. And I think that when we talk about modeling these, complex human conditions in an animal, it's very important to refer back to the clinic to see if we can recapitulate key aspects of what we see there in our system. And for the purpose of this talk, the most important phenotype is the excitation inhibition phenotype, which manifests as seizures in the clinic, present in over 85% of patients. And in our zebrafish model, we see this as a change in cell numbers. So the heterozygous mutant has a decrease in the number of inhibitory interneurons and a slight increase in the number of excitatory neurons, which shifts EI towards excitement. We also confirmed that there was a commissural deficit by looking at the anterior commissure and a proliferation phenotype, which reflects the microcephaly that we see in patients. So having established that the model is valid, I next wanted to use it as a tool to investigate disease mechanisms, for which I was fortunate enough to get an antibody to work in the fish. It's never quite straightforward with the zebrafish. But along with my excitement at having this new tool to play with came what can be described as existential dread when I saw what looked to be a specific peptide signal in what we would presume to be a null fish. But after calming down a bit and looking again at the FOXG1 coding region, I found that after this premature termination codon, we actually have 10 ATGs in frame with the wild type stop. And because the antibody I use targets down here at the C terminus, if there's a reinitiation of translation from any of these, that would be picked up. So I set off on a very long and tiring Western blot journey, but I was indeed able to show that the heterozygous and homozygous mutants had this smaller protein band of around 35 kilodaltons, which actually corresponds to reinitiation of translation from one of these cryptic start codons. And although that might be interesting in its own right, it doesn't really mean anything unless it does something relevant to disease. So to answer this question, I first of all overexpressed the mutant transcript in wild-type fish and found there was this sort of dose-response increase in the number of TBR2 positive excitatory cells, which is sort of reminiscent of the phenotype that I did during the characterization. Then on the other hand, I found that when I inhibited translation from this cryptic start codon using a morpholino, it rescued the excessive number of TBR2 positive cells, which suggests that the presence of this N-terminally truncated protein might actually contribute to the phenotype in heterozygous mutants and highlights it as a novel therapeutic target for FOXG1 syndrome. So you may have noticed when I showed these blots before that we have these smaller bands in the C-terminal and the N-terminal blots. And although this was a bit puzzling at first, I soon realized that if you add the size of the smaller band in the N-terminus to the C-terminus, it gives the size of full-length FOXG1. And that fit quite nicely with a previous report that showed FOXG1 is able to undergo a post-translational cleavage event with this C-terminal fraction, equivalent to this green domain up here, able to localize to the mitochondria. And because our mutant peptide will contain that domain, I thought, okay, well, maybe there's a mitochondrial phenotype in our mutants. For this, I started with just some simple immunostaining for FOXG1 and COX4. And when I quantified the structural phenotype of the mitochondria, I found that the mutants had not only an increased number, but those mitochondria were also increased in size. And this was more apparent in the dorsal telencephalon, which is the anatomical location of the TBR2 positive cells. So where we think this peptide is having its effect. Then when I treated with the morpholino, I first of all saw that the peptide signal was completely abolished in the homozygous mutant, which was reassuring. And I also found that the mitochondrial structural phenotype was rescued. And to be sure that this was sort of dependent on the increased mutant peptide, I looked at the co-localization of FOXG1 with COX4 and found that under control circumstances that was increased in the heterozygous and homozygous mutant and was rescued with the morpholino again. The structural phenotype suggested that there might be a functional mitochondrial phenotype. So for this, I used the seahorse machine from Agilent to perform a mitochondrial stress test. Essentially, you measure the oxygen consumption rate of individual embryos when you challenge the electron transport chain with different drugs. The main take-home message from these experiments was that the mutants again had increased ATP-coupled respiration and oxidative phosphorylation, which was again rescuable with addition of the morpholino. But what is FOXG1 really doing in the mitochondria? So for this, I was fortunate to have some RNA sequencing data. And here you can see the enriched gene ontology terms that came out of that experiment. In particular, these two jumped out of me, so RNA metabolic processes. This, combined with a recent report from the Malamachi lab, which showed FOXG1 is able to regulate cytosolic translation, got me thinking that perhaps FOXG1 might also be acting as a regulator of mitochondrial translation. We know that the mitochondria contain their own genome. This genome has 13 protein coding genes, all of which are members of the electron transport chain, and they have to be transcribed and translated within that organelle. So I checked some potential candidates and found that when I looked at mitochondrial ribosomal protein S34, there was not only a huge increase in the fluorescence of this factor in their mutants, but also the area of the staining. And the co-localization with FOXG1 was significantly increased. Again, this was more apparent in the dorsal telencephalon, where the TBR2 positive cells are. So the current thinking was that this C-terminal mutant peptide was a sort of gain of function of the wild type peptide. So I next performed a co-IP for FOXG1 and mitochondrial ribosomal protein, and indeed found that the C-terminal fragment of FOXG1 does seem to be pulled down with the ribosomal protein, indicating that the two likely interact. And of course, that's all well and good, but zebrafish tend not to get FOXG1 syndrome, so I wanted to make sure that this was translatable to human patients. For this, I was fortunate enough to receive an iPSC line from a FOXG1 syndrome patient that has a mutation very similar to our zebrafish model, with these cryptic start codons conserved from fish to mouse to human. And when I repeated my Westerns, I did indeed see that the FOXG1 syndrome line again had this smaller peptide band, which corresponds to reinitiation of translation from one of these cryptic start codons. I next wanted to see if this resulted in a mitochondrial phenotype. And for this, I looked at day in vitro 40 neurons because that was where the phenotype was most apparent in the zebrafish. And again, found that the FOXG1 syndrome line had an increased mitochondrial footprint and branch length, just as we saw in the zebrafish study. And there was a trend to an increased percentage of FOXG1 overlapping with COX4. So in summary, from the wild type allele, translation happens as normal, and we have this full-length FOXG1 peptide that is able to be cleaved with the C-terminal fragment able to go to the mitochondria. From the mutant allele, the premature termination codon drives the translation machinery further downstream, producing only the C FOXG1 mute, which is transported to the mitochondria at a higher frequency, and its increased abundance within the organelle at earlier time points leads to a precocious increase in the interaction between mitochondrial ribosomal proteins and FOXG1. And although I didn't have time to show this today, we also have some evidence that this causes an increase in the translation of mitochondrial transcripts, which in turn shifts the cell's energy production to oxidative phosphorylation. And with that, I would like to say thank you to everyone in Corinne's lab and to you guys for all listening.
Thank you so much. Thank you so much, Hannah. Are there any questions for Hannah while I'm waiting for those to come up, if anybody has got any. So what is the sort of, what would you say is the key significance of the increase in oxidative phosphorylation versus, it looked like you were saying versus glycolysis almost.
Yes,
So
exactly.
I'm so sorry, but I mean, I really can't remember. I mean, it's horrific, isn't it? Just tell me the basics of you know,
Yeah.
of what that would really sort of
So
mean.
the reason that we find this so interesting is because we know that as neurogenesis progresses, cells switch their means of energy production. So when a cell is more stem-like, they rely on glycolysis. But when they differentiate and become more mature, they rely on oxidative phosphorylation. And in FOXG1 syndrome, we know that there's an early differentiation phenotype. We know as well that the mitochondria is not just a passive player in that role. It can actively cause cells to differentiate if you increase the oxidative phosphorylation capacity within a cell. That's recently been shown. I think Pierre van der Hagen's lab has done a lot of work to show that the mitochondria isn't just letting it happen, they're actively contributing to the differentiation process.
Very good. Thank you. That makes lots of sense. Thank you so much, Hannah.
No problem.
I don't know whether anybody else has got any questions. If not, there was one other thing I was interested in, which is a bit of a technical question, and I can't remember the details. You talked about doing a morpholino at one point and then looking for expression of the which marker was it?
The COX-4?
No, it was the T. Was it the TB?
TBR2, yes, so the neurons.
So how effective, what's the time that you're doing, how effective is the morpholino? At what time point are you doing that relative to the time that you're doing?
So the morpholino is injected at the 1 cell stage. And then the TBR2 quantification I've done at 24 and 48 hours. So the morpholino hangs around for quite a long time. I think up to 2 to 3 days. It was tested to make sure it works, obviously, in the homozygous mutant. So you can see when we treat with the morpholino, it completely gets rid of the peptide signal from the homozygous mutant, which suggests that, you know, it's working effectively.
Lovely. There is one more question, so I'll just have this one more. Catherine Brown, nice talk. What's the link between the mitochondrial phenotype you see and the change in proportion of excitatory versus inhibitory neurons?
Yes, that is the big question. So in kind of my thinking, there seems to be a sort of dorsal-ventral. So we know FOXG1 is differentially regulated along the dorsal-ventral axis. It's higher expressed in the ventral part of the telencephalon and lower in the dorsal part, which is where these TBR2 positive cells are. The honest answer at the moment is we don't know why it's specific to TBR2 cells, the mitochondrial phenotype. But the reason we think we don't see a difference for the DLX neurons is because we think that requires the full length activity of FOXG1. So it's transcription factor role. And so that's why we don't see a rescue in the DLX interneuron number when we treat with the morpholino. We only see the rescue for the TBR2 positive cells. So there's a kind of full length versus cleaved peptide, different things going on, you know.